A very warm hello to all my lovely students. I hope you all are absolutely fine and studying very hard. So what we were talking yesterday was about amino acids and the essential amino acids, the non-essential amino acids and the conditionally essential amino acids. So let's take a quick recap in which we will talk about the 20 types of amino acids. As you can see, this is where we left. So the essential amino acids were the PVT, TIM and HLL, that is your phenylalanine, valine, tryptophan, then threonine, isoleucine, methionine, histidine, lysine and leucine, right? So these were the 9 essential amino acids which were very important and they cannot be made in the body, so they are required from the diet that we eat. The other non-essential amino acids are the other essential amino acids which are important but they are synthesized by the body and may not be the requisite component of the diet. They are glycine, alanine, serine, cysteine, aspartate, asparagine, glutamate, glutamine, tyrosine and proline. And out of these non-essential amino acids are some which are conditionally essential amino acids. Especially this question comes that there are 2 amino acids, especially this arginine is important when it comes to an infant, but for an adult it becomes a non-essential amino acid. Okay, so just remember this, and then we move on to the digestion of proteins. How do we digest proteins? So in mouth, there is no protein digestion in the mouth. You know that only salivary amylase or ptyalin is there which digests a little bit of maltose. Okay, then mastication and lubrication of food takes place in the mouth, and then it breaks down into smaller particles called the bolus along with the saliva, and it moves into the stomach. Now in the stomach, the intestinal wall, what happens? The large protein molecules are broken down into small amino acids, okay? And the enzyme involved is the enzyme protease, okay? The enzyme protease is involved that breaks down large amino acids, large proteins into individual amino acids in the intestine. Now what happens when the food reaches the stomach, the amino acids or the proteins reaches the stomach? Now pepsinogen is the enzyme which converts pepsin from its inactive form to its active form. So pepsinogen is the inactive form, okay, and this is converted into pepsin, which is the active form, because of the acidic pH of the stomach and because of the presence of HCl. So pepsinogen is converted into pepsin, which is the active form, and then pepsinogen pepsin acts on the proteins and amino acids to digest them and to form polypeptides. Once the acidic chyme it reaches the small intestine, the pepsin activity now completely stops. Now the chyme mixes with the alkaline pancreatic juice in the duodenum and also in the jejunum. Now what happens in the small intestine? The polypeptides which are formed are now acted upon by enzymes like trypsin, chymotrypsin and elastase. So there are 3 main enzymes: trypsin, chymotrypsin and elastases, which cleave or break the peptide bonds within the peptide chain, and therefore these enzymes are also known as endopeptidases. They are the ones which cleave the peptide bonds of the amino acid chain. And now what happens? There is another enzyme called the carboxypeptidase, and this hydrolyzes the bond. Okay, this hydrolyzes the this carboxypeptidase, basically it is released from the pancreas, and the aminopeptidases of the intestinal wall are the exopeptidases. So trypsin, chymotrypsin and elastase are the endopeptidases because they cleave the peptide bond within the polypeptide chain, and carboxypeptidase is released from the pancreas. The pancreas, remember, the ones that release the insulin. So from the pancreas, and carboxypeptidase is an exopeptidase because it is coming from outside, and also the aminopeptidase, they are coming from the intestinal wall. So they are known as the exopeptidases. Okay, so you remember that they are known as the exopeptidases. Now carboxypeptidase, what it does, it hydrolyzes the bond at the carboxyl terminal, of the chain of the amino acid, and it breaks the amino acid. Now some free amino acids are released, and these free amino acids are released in the lumen of the small intestine, and some of the other amino acids are released at the intestinal wall surface by the action of those aminopeptidases and dipeptidases. So now dipeptidases, what do? They cleave the dipeptides into their individual single amino acids, and this is how amino acids are formed. After this, what happens? Absorption of amino acids occurs, and they are absorbed in the same way that carbohydrates was absorbed by the microvilli of the intestinal cell wall. Okay, they are absorbed into the intestinal cell wall, and then they are there in the bloodstream. Now next we talk about the transport of amino acids. What happens? How is the amino acid transported? Some transport mechanisms are there which help in the transport of amino acids. Okay, like sodium is there for glucose. Sodium is there which helps in the transport of amino acids. Similarly, in the same way, sodium is there which helps in the transport of carbohydrates or glucose. Sodium is there which helps in the transport of glucose. Similarly here, the amino acids are also transported with the help of sodium. So you know, when a person is having a low glucose in his body, or maybe he's having low sugar in his body, so they give a lot of electrolytes, they give lemon, they give water, they give salt along with it, so that if sodium is not present properly in the body, it will not be able to transport the glucose molecules from the intestine to the bloodstream and to the cells where it gives energy. So similarly, the same case happens with proteins also. If the sodium molecules are not there in proper amount, the amino acids will not be able to be transported. There are also some other independent carriers. I mean, they are not dependent on sodium. So transport of amino acid, first maybe along with sodium, like the glucose transport mechanism, and the others may be sodium independent carriers. And which are these sodium independent carriers? They are, one, the basic amino acids are there. These are carried by the sodium independent amino acid carriers, and the aliphatic amino acid carriers are also sodium independent. So they carry them without the help of sodium ions. Now from the intestinal cells, the amino acids pass into the blood capillaries that lead it to the portal vein, that is the vein that goes to the liver, and this vein, through the blood, it goes to the liver, and the concentration of amino acids in the portal vein rises soon after ingestion of a high protein diet, and once they reach the liver, they are synthesized into plasma proteins. Most of them are synthesized into the plasma proteins, and the remainder is sent via the blood to the individual cells for synthesis of specific proteins, like structural proteins, like transport proteins, like keratin, whatever we had talked about in the last class. Now let's talk about the factors affecting the bioavailability. The bioavailability. Now what does the term bioavailability mean? See, the bioavailability means to include a single concept, the effect of a sequence of metabolic events, like after digestion, after solubilization, after absorption, after the organ uptakes, how much available protein is there for the body, and also after the mechanism of secretion, after the mechanism of excretion, how much of protein that we've eaten is available for the body. So it basically, bioavailability gauges the extent to which any administered substance, maybe protein or anything, reaches its site of action or utilization and in the body. So it is the measure of the efficiency, how much of what is ingested is actually used. Like if you're taking a lot of protein, but the protein is not getting digested, it is coming out through the excretion just like the way it has been taken in, that means that the body is not being able to uptake the protein. Only if you've taken 100 grams of protein and only 20 grams of protein are there, which the body is using up. So how do we know how much of what is ingested, how much protein ingested is actually used for its intended purpose? And for that we basically talk about the bioavailability of proteins. Okay, so and when we talk about the bioavailability of proteins, so basically see, when we talk about how the factors are affecting the amino acid bioavailability, so first factor we can say is by cooking. What happens in cooking? When we cook the food, the proteins or the amino acids are more or less, they are sensitive to heat. Like arginine is very stable and only decomposes when it is, kept on temperatures above 470 degrees Fahrenheit. So arginine is heat stable, but carnitine, it decomposes at a temperature of say around 284 degrees Fahrenheit. So cooking, when we cook, it does kill the microorganisms in many non-vegetarian food products, but it also makes the long spiral polypeptide chains unwind. The polypeptide chains are like this, so they unwind, okay, and it causes the amino acid to become more exposed when it reaches the digestive system. Second thing that we can talk about here after cooking is the nature of the food, or we can say it, the physical nature of food, whether solid, liquid, powder, tablet, what extent it has been chemically pre-digested. That means it is easily absorbed in the body, and the type of amount of binders that has been added to it, of fillers or maybe some other nutritive or non-nutritive materials that have been added, that affects the bioavailability. Maybe say if we say for example, if we take vitamin C tablets with lemon, so it increases the bioavailability of that iron. Okay, if we take iron capsules with lemon, it increases the bioavailability of vitamin C. It increases the absorption of iron, and vice versa. Iron helps, it is absorbed properly. The non-heme iron is absorbed properly just in the presence of vitamin C. So what is happening? The vitamin C is present and it increases the bioavailability. Similarly here, the physical nature of food, the amount of non-nutritive materials that have been added or the nutritive materials that have been added changes the absorption or the digestion rate of the food system. Okay, then the status of the digestive system, like no matter how much protein you are eating, but your body refuses to digest it. And especially genetics plays a very important role. So if maybe it is running in the family that you are suffering from an irritable bowel syndrome, or if you're suffering from some kind of a problem that your proteins are not getting digested properly and absorbed properly, so they won't, right, no matter how much protein you eat. And last but not the least, it also depends on the metabolism. How does it depend on the metabolism? See, its utilization before absorption, that is, its utilization in the intestine before absorption, and this something that occurs with glutamine, like metabolism or utilization in the liver occurs, and for like we can say for maximal effects, the amino acids should be taken on an empty stomach, and so that it can reach the target tissues. So we can say that the metabolism also before absorption, what the metabolic rate is of a person, that also determines the amino acid bioavailability. Okay, and they say that if amino acids are taken on an empty stomach, it reaches the liver and then it reaches the target tissues very easily. Okay,